Assalamualaikum and very good morning. A warm welcome to all participants from Malaysia, the SEAMEO network and affiliated countries to IMR-SEAMEO webinar series 2022. Our theme today is Ending the Diagnostic Odyssey for Inborn Errors of Immunity using Whole Exome Sequencing. Our presenters today are from the Institutes for Medical Research in Malaysia. In conjunction with World Primary Immunodeficiency Week started today, we join the efforts of organizations across the globe to promote awareness of primary immunodeficiency diseases, or more commonly now known as inborn errors of immunity. I am Dr. Adiratna Matripin, the head of Primary Immunodeficiency Unit, Allergy and Immunology Research Center, IMR, will be the moderator for today's session. Ladies and gentlemen, this webinar series was initiated with the aim in mind to share the knowledge, discuss topics on biomedical research, and to present research outcomes. Before we begin our presentation, allow me to announce a few housekeeping messages from the Secretariat. Number one, we will not be taking questions during the presentation, but participants may post their questions using the link that have been shared in the invitation email and YouTube channel. Number two, the question and answer session will be conducted at the end of the second presentation. Number three, the attendance link is also provided for those who would like to receive an e-certificate for attendance. You may find the link in the chat box of the YouTube channel. Ladies and gentlemen, for today's webinar, we have two speakers. Our first speaker is Dr. Mohd Farid, who is a medical officer in PID unit under Allergy and Immunology Research Center, IMR. He holds a medical degree from the University Science Malaysia. His research area of interest are genetics and cellular defects of inborn errors of immunity. He has published numerous papers in international and peer-reviewed journals. For today, Dr. Farid will be delivering a presentation entitled Overview of Inborn Errors of Immunity, Clinical Presentation and Diagnostic Challenges. Dr. Farid, the screen is yours.
Thank you, Dr. Adiratna, for the kind introduction. A very good morning and Assalamualaikum to all. I'll be giving an overview of inborn errors of immunity, the presentation, as well as the challenges that we face in our setting. These are my presentation outline. The immune system is a complex system comprising of numerous cells in the body, together with numerous humoral components, and it works in different organs in the body, where the main function of the immune system can be summarized into two. So the first is actually to fight invaders, where the aim is actually to protect the body, and number two is actually to ignore anything else. Anything else refers to our host antigen. It is very important for the human immune system not to mount any reaction to the host tissue, otherwise auto-inflammatory will set in. Inborn errors of immunity is a group of more than 400 genetically characterized diseases. So the manifestation of this condition includes increased predisposition to infection, auto-inflammatory diseases, allergy, and malignancy. This condition was previously known as primary immunodeficiency, and even until now, both the terminologies IEI and PID are used interchangeably. IEI is due to monogenic defect affecting genes involved in the immune system, and it was once considered as a rare disorder. However, the actual incidence is not clearly defined. The list of genes involved in IEI are continually being updated. So there is actually a society which is responsible to overlook this. So the name of the society is the International Union of Immunological Societies Expert Committee of Inborn Errors of Immunity. So every 2 years or so there is update, and usually what we do is we refer to the latest update during our analysis. This figure here illustrates the ever-expanding gene list in inborn errors of immunity between the year 1983 to 2019. As we can see that since the past few years, the number is increasing rather quite exponentially. This is the current classification of IEI. IEI are being classified into 10 tables, and the lists are here. how do clinicians suspect that there's any possibilities of IEI? So IEI, the clinical manifestations include predisposition to infection, autoimmunity, malignancy, allergy, and inflammation. There is this chart that has been used by clinicians worldwide in order for them to diagnose IEI. So this is actually being produced by Jeffrey Model Foundation, which is a non-profit organization, the main role of this organization is to increase awareness on primary immunodeficiency. So when we look at this figure, we suspect PID, especially when a person has 4 or more new ear infection, they have 2 or more serious sinus infections every year, and etc. When we deal with cases of immune errors of immunity, it follows the usual workflow. First, a detailed history taking is important, especially family history. Second, we perform a preliminary investigations which include the differential counts, blood culture sensitivity, and also immunoglobulin and complement levels. Next, we move on to functional study, and usually what we do is we perform flow cytometry or any other form of investigations to look at the protein expression, and this is followed by a confirmatory test using genetic study. So it can be a single gene study, or it can be a polygenic study using next generation sequencing. For the functional study, flow cytometry is a very important tool in the diagnosis of inborn errors of immunity. So the first example that I'm giving you here is that the use of flow cytometry in the detection of BTK protein, which is the protein responsible for X-linked A gamma globulinemia. Look at this purple colored histogram. So in a normal person, there is normal expression of the BTK protein. In a patient, there is deficient expression of the protein, as well as the mother who is a carrier. She actually shows bimodal expression. So bear in mind that BTK is inherited through X-linked mode of inheritance. Moving on to the confirmatory test using genetic study, the first or the earliest method rely much on the Sanger sequencing. This is very useful when the condition is when there is a clear genotype-phenotype correlation. For instance, we have a male child presenting with early onset infection. When we perform preliminary studies, the patient has low B cells, hypogamma globulinemia, and then we can perform BTK protein. However, due to the very broad genetic background of IEI, and sometimes there is a great overlap between the genes, next generation sequencing now is actually the current preferred method because it allows simultaneous screening on multiple genes. So this is divided into two, which is study of the exome, we call it as WES, or study of the whole genome, we call it as WGS. Now I move on to the diagnostic challenges that we face in our lab. So the first one is actually genetic or disease heterogeneity. The first one is one gene can present with many phenotypes. An example here will be a STAT3 gene. So this is actually a gene which is responsible in transcription factors. So in the event of gain of function condition, patients manifest with lymphoproliferative syndrome. However, when the STAT3 manifests in loss of function feature, patient typically presents with autosomal dominant hyperimmunoglobulin E. Another example would be STAT1. When we have loss of function, patients develop susceptibility to mycobacteria and salmonella infection. However, in cases of STAT2 gain of function, patient manifests with chronic mucocutaneous candidiasis. Second challenge is that one phenotype can be shared by many genes, meaning that defects in multiple genes presents with one common phenotype. So such as, number one, race IgE. This can be seen not only in STAT3 mutation, it can also be seen in DOC8 mutation, TYK2 mutation, WAS mutations, and few more. And second example would be the occurrence of hemophagocytic lymphohistiocytosis, or HLH, which can be seen in patients with Baharin mutation, syntaxing mutation, or ZIAP mutation. Another challenge that we face here is actually the need for a very elaborate and extensive screening and confirmatory tests. Most of the tests are not widely available. for instance, flow cytometry and Sanger sequencing, or even next generation sequencing. Even when it is available, it can be very, very costly, especially flow cytometry reagents and next generation sequencing. For flow cytometry, for instance, there is a very stringent sample requirement because we are measuring the expression of protein on cells. So we need the cells to be transported on certain temperature at certain timing. So it is very, very crucial. And then when it comes to interpretation of laboratory reports, some medications that patients are receiving may interfere with the laboratory reports. And the last is that when NGS is available, it is compounded with the need for special facility as well as specialty to perform the diagnosis, to perform the analysis. And also another important area is that there is a lack of database that actually represents the genetic data of our local population. I'd like to end my presentation today with 3 take-home messages. Number 1, inborn errors of immunity has a broad clinical manifestation and diverse genetic background. We have seen earlier that one gene can manifest with many clinical phenotypes, and one clinical phenotypes can be shared by defects in multiple genes. The second take-home message is that in order for us to suspect if a patient has primary immunodeficiency, we can always refer to the chart given by the Jeffrey Model Foundation, which is the 10 warning signs of primary immunodeficiency or inborn errors of immunity. And number 3, the laboratory diagnostic for IEI is complex because it requires an elaborate preliminary as well as confirmatory studies, and even the data interpretation is quite complicated. The materials from my presentation today are derived from these articles. I'll be more than happy to answer questions at the end of the webinar. So for now, I'll pass over the screen to Dr. Adirana. Thank you so much for your attention.
Thank you, Dr. Farid, for the interesting talk. If there are any questions that you would like to ask about the presentation, kindly submit your questions using the link provided in YouTube channel. Our second speaker is Ms. Chear, Teng, who is a research officer in PID unit under Allergy and Immunology Research Center, IMR. She holds a degree in Master of Science in Genetics and Molecular Biology from the University of Malaya. She's completing her doctoral degree in Bioinformatics from the same university. Her research area of interest are genetics, biochemistry, and molecular biology in inborn errors of immunity. She has published many papers in international peer-reviewed journals. It is our great privilege to have Ms. Chear with us to share her research. Her presentation is entitled Utility of Whole Exome Sequencing in Molecular Diagnosis of Inborn Errors of Immunity. Over to you, Ms. Chear. The screen is yours.
Thank you, Dr. Adi, for your kind introduction. Thanks the organizer for giving me the opportunity to give a talk in today's webinar. Good morning, everyone. I am Chear Teng. Today, I'm going to talk about utility of whole exome sequencing in molecular diagnosis of inborn errors of immunity. This is the outline of my presentation. I will briefly talk about inborn errors of immunity, challenges in disease diagnosis, whole exome sequencing, and followed by a sharing of our research findings from a case study. Please allow me to introduce again on inborn errors of immunity, IEI. IEI was previously known as primary immunodeficiency, PID. IEI are genetic disorders in which part of the body's immune system is missing or does not function properly. Considering defective immune response, IEI patients are prone to get infections, allergy, malignancy, autoimmunity, and/or autoinflammation. These pictures show the clinical presentations in some IEI patients. To date, more than 500 conditions have been described to be arising from more than 400 genes related to immunity. To identify the nature of an illness, clinical diagnosis can be made based on clinical signs and symptoms, patient's family history, along with physical examinations and laboratory diagnostic tests. To obtain an accurate molecular diagnosis in genetic disease, amplification of candidate genes using polymerase chain reaction (PCR) followed by Sanger sequencing are essential to identify the DNA mutation responsible for the disease. A disease diagnosis remains difficult if solely based on the clinical phenotypes. When common symptoms are shared among different kinds of diseases, or the patient may exhibit complex or atypical clinical phenotypes. The disease diagnosis becomes challenging when the disease is caused by mutation in multiple genes or variants. Because of these issues, a diagnostic delay of about 4 years have been observed in Malaysia IEI cohort, and this diagnostic delay has resulted in an organ damage and or mortality in the patient. To prevent this serious clinical outcome in the patients, an early and accurate molecular diagnosis is important. Sanger sequencing is gold standard in molecular diagnosis. However, sequencing of multiple candidate genes appears laborious, costly, and time-consuming. Therefore, whole exome sequencing have been an alternative in rare genetic disease diagnosis. Although human genome consists of approximately 3 billion nucleotides, 1 or 2% of the human genome is translated into functional protein. It was described that 85% of the disease-causing mutations occur in the protein coding region. Whole exome sequencing allows sequencing up to 20,000 protein coding genes simultaneously, appears a cost and time-effective strategy as compared to Sanger sequencing. Whole exome sequencing has been widely used in the molecular diagnosis of genetic disorder such as IEI with complex clinical presentations. The objective of this presentation is to demonstrate the usefulness of whole exome sequencing in diagnosing inborn errors of immunity (IEI). This slide shows several publications from our lab. In this presentation, I will share with you the research finding from one of the published papers entitled "A novel de novo NLRC4 mutation reinforces the likely pathogenicity of specific LRR domain mutation." The study subject was a Malay girl born from non-consanguineous parents. She started to have sepsis when she was 2 weeks old, followed by recurrent soft tissue and skin abscesses. Since 2 years old, she had recurrent prolonged fever. When she was 5 years old, she had abdominal pain associated with fever and vomiting. However, no microbe was isolated from pus and blood culture. When she was 12 years old, she had right knee inflammatory arthritis. But the immunological investigation were normal. Considering recurrent prolonged fever and inflammation, in the absence of infection and family history, she was suspected to have inborn errors of immunity related autoinflammatory disorder. However, this type of disease was described to be associated with more than 40 genes. To obtain an accurate molecular diagnosis for this patient, she was recruited in this study. Let's move on to the methodology. Genomic DNA was extracted from the peripheral blood mononuclear cells from the patients. This DNA was subjected to whole exome sequencing. Millions of short reads were generated and then were subjected to subsequent bioinformatics analysis, such as read alignments to the human reference genome, followed by variant calling to identify the variants present in the sequencing data, and then variant annotation and effect prediction. To identify the causative gene mutation in the patient, variant prioritization was performed. by multiple step filtering strategy. The candidate variant that is shortlisted from this analysis were then subjected to variant validation by Sanger sequencing. To understand the functional impact of the mutation, functional assay was performed, such as cytokine measurement in this study. Let's move on to the result part. Whole exome sequencing generated 52 million pair end reads with 150 base pair length from each end. Of these reads, 98.89% mapped to human reference genome. Variant analysis showed that the patient had more than 23,000 exonic single nucleotide variations in 9,360 genes, and also 701 exonic indels in 525 genes. Following stringent multiple step filtering strategy, we shortlisted a potential mutation in the NLRC4 gene. This mutation is a missense mutation, which involves the nucleotide change from A to T at the position 1970 nucleotide, which leads to an amino acid change from glutamine amino acid to leucine amino acid at the position of 657. This mutation was not reported in any population databases, indicating a novel mutation. Also, this variant was predicted to have deleterious and damaging effect as predicted by in silico pathogenicity prediction tools. Sanger sequencing confirmed the heterozygous mutation in the patient, as highlighted in this arrow here, while the parents did not carry the mutant alleles. This result shows that the patient had a de novo mutation. Multiple sequence alignment of NLRC4 protein shows that the glutamine amino acid is evolutionarily conserved across several species. Therefore, substitution of leucine amino acid at this position may disrupt the protein function and also its structure. Let's look at the NLRC4 protein. NLRC4 protein play a role in host defense against gram-negative bacterial infection. Following bacterial infections, NLRC4 proteins are activated to form a complex called NLRC4 inflammasomes. Activated NLRC4 inflammasome can initiate the downstream caspase-1 activation, which leads to inflammatory cell death and also production of pro-inflammatory cytokines, which then leads to inflammation. In the absence of bacterial infection, mutation in NLRC4 can cause inflammasome hyperactivation and leads to inflammation. Excessive interleukin-18 production is a hallmark for NLRC4-related autoinflammatory disorders. Cytokines analysis show increased interleukin-18 levels in patients with NLRC4 mutations. In this study, our study subject also had increased interleukin-18 levels in the serum as compared to the normal control. In summary, whole exome sequencing and bioinformatics analysis enable us to identify a novel pathogenic mutation in NLRC4, confirming the diagnosis in the patient. Cytokine analysis demonstrated increased interleukin-18 level consistent with other NLRC4 patients, supporting the pathogenicity. These results show how a point mutation in the protein coding sequence may affect its biological function. These are the references used in this presentation. I would like to take this opportunity to thank Ministry of Health Malaysia for funding this study, A special thanks to co-investigators from hospitals, collaborators from University Malaya, as well as all staff from PID Unit, Allergy and Immunology Research Center, IMR, for their contributions. With that, thank you for listening.
Thank you, Ms. Chear, for the insightful presentation. Here, I would like to briefly summarize that it is a diagnostic challenge to diagnose inborn errors of immunity due to diverse presentations in patients, which can also be unique in our population due to multi-ethnicity and climate difference. It is important to detect this group of patients and provide a definite diagnosis in order to manage them accordingly and provide them with optimum treatment. We will now moving to questions and answer sessions. Please do not hesitate to send us your questions, and I will try my best to connect you to our distinguished speakers through the posted questions. Nevertheless, if time does not allow us to address all the posted questions, our speakers will try their best to answer through email. So it seems that we have received a few questions from our audience. Question number 1. Why is genetic diagnosis important for inborn errors of immunity? Dr. Farid, hopefully can answer this question.
Thank you, Dr. Adi. So this is indeed a very important question. Establishing genetic diagnosis for IEI is important because it determines what kind of treatment patients will receive. So for instance, in the cases of patients with Bruton tyrosine kinase deficiency, this is actually a gene which is responsible in B cells maturation. So these patients, they lack immunoglobulin. If the treatment will be infusion of immunoglobulin at regular intervals, 3 weekly, 2 weekly, or monthly, and they do well with the infusion. Another extreme example would be in cases of severe combined immunodeficiency, such as in cases of JAK3 or REC1 or REC2 mutation. Because this is so severe, they manifest very early in life, and then the definitive therapy for these people, for these individuals, will be stem cell or bone marrow transplantation. Another form of severe combined immunodeficiency, which is ADA deficiency, they lack a specific enzyme. They do well if they receive enzyme replacement. And the last example that I would like to give is in the cases of chronic granulomatous diseases, for instance. When we diagnose somebody has a CGD associated genes, sometimes the carrier mother may manifest with symptoms, which is commonly GI granuloma or GI manifestation. And to sum up, the genetic diagnosis in IEI is also important because we know that it is a genetic disease. There's always chances that the future offspring might be affected. So it is very, very important for family counseling. I think that's all I have. Back to you, Dr. Adi.
Thank you. Thank you, Dr. Farid. We have some more questions, I suppose. There's one more. What is sequencing library and what is short read in sequencing? Ms. Chear, can you answer this question?
Thank you, Dr. Adi, for the question. What is sequencing library? Sequencing library is actually a pool of DNA fragment that is generated and attached with the adapters in whole exome sequencing experiment. And this sequencing library is important so that the DNA fragment is compatible to the sequencing platform that we are using to sequence the DNA fragment in whole exome sequencing. For example, in Illumina platform, the DNA will be fragmentized into DNA fragments in various sizes, and then the DNA fragment will be then ligated with adapters, and these adapters sequence are complementary to the sequence that sequence of the oligonucleotide that is present on the floor cells of the sequencing platform. Okay, then this library, sequencing library that is loaded on the floor cell will then subjected into next generation sequencer to sequence the DNA fragments. Another question is what is short reads in sequencing? Generally, short reads in sequencing just means the nucleotide sequence of a DNA fragment that is generated from NGS platform. I think that's all from me. Back to you, Dr. Adi.
Thank you, Ms. Chear. So we have numerous questions here. So, okay. So there's a question to Dr. Farid. How can clinicians send genetic study for IEI in Malaysia? From Dr. Rizwan, USM.
Thank you for the question. If you have any patients you suspect to have inborn errors of immunity, you can contact us in IMR and we can actually arrange a session for your patient. Thank you.
So basically, this to answer Dr. Rizwan, we are conducting this research still until 2023. So if you have any samples or if you have any possible candidate, do contact us and email us, and we will approach you for the details on how to send the samples. All right. So next question to Ms. Chear. In the event where unusual mutation is seen in Sanger sequencing analysis, how to differentiate IEI mutation from mismatches that arise due to the polymerase misincorporation of bases? Ms. Chear?
Okay, thank you, Dr. Adi. Thank you for this question. For whole exome sequencing, because we are doing whole exome sequencing, we will have to have a very high coverage, sequencing coverage to cover this issue. When you have a mismatch at a certain position, when you have a very high coverage of this sequencing coverage in this from this whole exome sequencing, then you will be more confident to call it as a mismatch. And then because for whole exome sequencing, which involve the cluster generation, it will have some it will lead to some sequencing errors. So to overcome this issue, normally we will validate this single nucleotide variation, for example, by using Sanger sequencing, which has more sensitivity in detecting mutation. I think that's all from me. Thank you, Dr.
Thank you, Ms. Chear. Next question for Dr. Farid. Do you have the data on prevalence of inborn errors of immunity in Malaysia?
Thank you for the question. I do not have the correct number with me at the moment, but there are emerging reports describing certain IEI conditions in Malaysia, such as BTK cases. We have quite a few. We have quite a few publications on chronic granulomatous diseases, and also some other IEI conditions. I'll get back to you with the numbers that I derive from these sources. Thank you.
Thank you, Dr. Farid. Actually, we do not have a clear prevalence of inborn errors of immunity in Malaysia, but we do have a database. And what we have so far, when we translate to the prevalence, it's actually very, very low, meaning that it is underdiagnosed, or perhaps the awareness of this disease and it's not there yet. So perhaps that's the reason why we are not detecting more cases like any other countries. So that's how we can, that is one of the reasons why we are trying to collect and to do this study to at least to confirm that this is actually not very rare and is happening in Malaysia. So that's how. Thank you, Dr. Anna Sufiza, for the question. Next question. This is a question for Ms. Chear from Dr. Huang. What type of samples, what tubes, containers, preservations and transport, for example, for this test, especially in Sabah Sarawak region? And is postmortem samples valid for investigation? Ms. Chear?
Okay, thank you, Doctor. Thank you, Mr. Wang, for this question. What type of samples that we need for this whole exome sequencing is basically blood samples from live patients. For whole blood, normally we'll collect the blood in ETDA tube, and then you just transport in normal room temperature to us. But before that, to request for this test, because this is still under research basis, so if you need to send this test to us, then you need to contact us, contact our team. For example, you have to talk to Dr. Adi regarding this request. And then as for your second question, is postmortem samples are valid for this investigation, right? The second question is that the answer for this question is that as long as the samples contains DNA, I think it should be valid for investigation, as long as the volume, the concentration for the DNA that extracted from these postmortem samples sufficient for the whole exome sequencing, then I think it's valid. For example, we need about one microgram of the DNA concentration to be sent for whole exome sequencing. Okay, thank you.
All right, thank you, Ms. Chear. So there's another question for Dr. Farid. What is the next step after the diagnosis, and what is the next step for the patient's management?
Thank you for the question. We receive samples in multiple packages. Multiple packages means that sometimes you only receive patient sample, sometimes you receive the family sample, the father and mother, sometimes only the mother, sometimes only the father. So usually what we do is actually upon establishing of a genetic diagnosis for a patient, our first priority is actually to look on the parents, because we wanted to see are they affected or are they contributing anything to the disease in the patient. And subsequently, we are requested to perform subsequent studies on the family members. Sometimes we also do functional study, because what's so special about genetic diseases is that even in the presence of mutation, sometimes the protein expression or the protein function is normal or reduced or even absent. So if we manage to develop any downstream investigations to support the pathogenicity of the mutation, we will perform, which is mostly flow cytometry. We'll obtain antibodies specific to certain proteins, and we perform that on samples, patient samples as well as parent sample. And yeah, I think that's all. Thank you.
Yeah, I think probably the person who asked the question would want to know about the management of the patient. For example, what do we do after the diagnosis, and what are the managements that we can provide for the patients?
Okay, all right. Sorry for missing that part. So upon diagnosis, it is very important that our IEI patients, because of their increased predisposition to infectious diseases, they must be covered with prophylaxis antibiotics, which includes even antibacterial, antifungal sometimes. And then with these patients, they need to be under the care of immunologists, or most of the time also by the infectious diseases specialists, where their therapy is actually a bit different from other people. When they receive vaccines, some people or some IEI patients, they cannot receive live vaccines, for instance, and then they also require regular screening on certain metabolites, for instance. Another example will be in cases of CGD, the mothers, if the mother is a carrier, they can develop symptoms as well. So we should anticipate symptoms. And another example I would like to give is actually in the event of STAT3 mutation or Wiskott-Aldrich syndrome mutation. Any individuals with these two mutations, we know that sooner or later they might develop any form of malignancy, and therefore special care needs to be taken, so that we should always look for any sign and symptoms of malignancy in these patients. Thank you.
Thank you, Dr. Farid. Question for Ms. Chear. How transcriptome sequencing is different from whole exome sequencing, and can it be used to diagnose inborn errors of immunity? Ms. Chear?
Okay, thank you, Dr. Adi. Thank you for this question. Transcriptome sequencing is basically look at the expression level of the RNA level, for example. And for whole exome sequencing is basically a sequence of the whole exome of the genome to investigate if there is any gene mutation at specific location. So for in my opinion, for these two platforms, it used to either check the expression level or the point mutation that occur in the samples. So I would think both of them can also use to diagnose the IEI. However, in normal scenario, we opt for whole exome sequencing to check for the gene mutation instead of transcriptomic profiling. Okay, thank you.
Thank you, Ms. Chear. Question for Dr. Farid. Do you know the mortality rate on patients with inborn errors of immunity in Malaysia? Question from Dr. Ana Sufiza.
Thank you, Dr. Ana Sufiza, for the important question, and it's quite a difficult to answer question. We do communicate with our collaborators regarding the outcome of all the patients that we diagnosed so far. A straight answer to your question is do you know the mortality rate on patients with IEI in Malaysia? Short answer is no. But based on our limited number of cohorts, we noticed that a certain diseases is associated with increased mortality rate. For instance, in our cohort of patients with hyper IgE due to STAT3 mutation, we have a few cases who actually have succumbed into the disease-related complication. We also have patients with severe combined immunodeficiency which succumb early, because we know that if they don't receive the definitive treatment of stem cell transplantation before the age 1, usually the prognosis is very, very poor. So I think in general, the mortality rate of patients with IEI in Malaysia, it depends so much on the type and the severity of the diseases. Some conditions like BTK, they are okay, they survive until almost normal. However, in certain cases which is so severe, such as severe combined immunodeficiency, the mortality rate is high, and these are the conditions which we believe that we have missed because they are not detected. Thank you, Dr. Adi.
Thank you, Dr. Farid. So another question is, could you briefly explain the general workflow for whole exome sequencing? Ms. Chear?
Thank you, Dr. Adi, for the question. For whole exome sequencing, generally it involves 4 steps. The first step is library preparation, whereby you will have to fragmentize your DNA into small fragments, and then it will ligate with the adapters, which is compatible to the sequencing platform. And then on the, for example, on the floor cell that the DNA library attached onto it, then the floor cell will be then subjected into the sequencing sequencer, and then on the sequencer itself, it will perform bridge amplification of the library, and then once it's perform the cluster generation, then you will have a lot of the similar strands on the floor cell. From there, you will have to perform the sequencing by before that you will have to have a step called exome enrichment, whereby you add all the primers that require for the whole exome sequencing. onto the system, and then you will get the output, which is the reads that you get, which is the sequence of the DNA fragment that is generated by the sequencer. And then for the analysis part, then you will need bioinformatics analysis for this data analysis. Thank you.
Perhaps, Ms. Chear, they would like to also understand the data analysis, the steps for the data analysis. Perhaps you can just summarize a little?
For bioinformatics analysis, once we got the raw reads from whole exome sequencing, then the first step is that we will check for the quality of the data, and then followed by reads alignment to human reference genome. For read alignments, it is like you play a jigsaw puzzles, whereby you have many pieces of a smaller piece of the puzzles that you need to make them into a complete picture. And then for example, the reads that you obtain is smaller fragments, then you will need to align and merge them back to the human reference genome, which is the big picture of the puzzles. Then from there, you will know each reads will map to which location, and then from there, you will know the alignment of the whole exome that you in your patient samples. And then after that, we will perform variant calling, whereby we will identify which is the single nucleotide variation that present in the sequencing data, or small insertion or deletion that occur in the data by this step. And then after that, we will perform variant annotation. For variant annotation is a process to provide functional information regarding the variant. So for this step, you will have information of the variant, for example, minor allele frequency, the functional impact of the mutation, as shown in my presentation slide just now. You will know if this mutation has been reported in any population databases before, or if the mutation is a damaging variant or is a benign variant, for example. So all this information can be obtained from this variant annotation step. And then after that, you have a list of the variants that you have analyzed by bioinformatics analysis. Then to identify which gene is the potential that causing the disease in the patient, then you will need to perform variant prioritization, for example. So in our study, we screen all the rare variants with minor allele frequency less than 0.001. That means it's a very rare variant, and also have damaging effects. And also the variant should have compatible clinical phenotypes with our patients. Then we will retain for analysis, for example. And then for the identified variant, then we will then perform a subsequent variant validation. And all these steps that I mentioned just now is for the bioinformatics analysis for the whole exome sequencing data.
Thank you. Thank you, Ms. Chear, for the elaborations and a clear explanation for that. So there's another question from Dr. Anas Fiza to Dr. Farid. Few European countries, USA and Korea, already started newborn screening for severe combined immunodeficiency. Would your group interested to explore a pilot project for SCID using RT-PCR?
Thank you for the question, Dr. Anas Fiza. So, developing newborn screening for PID, for instance, using RT-PCR, the Treg, quantification of the Treg from the dried blood spot for cases of SCID Of course, it is. Our team has actually discussed about this, and then we are interested in doing this. At the moment, we are at a preliminary discussion, because there are a few things that we need to look into the technology. We need to delve deeper into the false positive reports that can be derived, as well as who should be our target group for our pilot study. Even there was at one point we were thinking that it would better be requested as a dasar baharu rather than a pilot study, because we believe that in order for us to deliver this as a routine diagnostic is going to be quite the workload is going to be quite a lot, because we need laboratory technologies or trained personnel to extract the DNA. We also need officer levels to perform the elaborate interpretations of the report. And then, since our laboratory also has the ability to perform lymphocyte immunophenotyping, which can further strengthen the findings, we believe that a strong team, and a very big team is important. So at the moment, we are unsure whether this should be a pilot project, or this should be requested as a dasar baharu memperkasakan perkhidmatan diagnostik PID in Malaysia. Yes, we are interested in that, and we will continue on discussing and look at what is the most feasible steps to be taken by our team. Thank you.
Thank you, Dr. Farid. Actually, Dr. Anas Fiza, it is a very good question, and in order for us to actually launch this kind of project, we foresee that this is going to incur costs, but I think this is very important, just like newborn screening for inborn errors of metabolism. So we are thinking of potential collaborators and possible collaboration with SDC, for example, under Dr. Anas Fiza, because you are receiving samples from our babies to screen for inborn errors of metabolism. So there is a potential collaboration here. So we think that we should proceed, because this is important. If we are able to avoid or prevent severe combined immunodeficiency from setting in and develop into organ failure or organ damage, I think that is a good thing to start with. So thank you for the question, Dr. Anna. So there is some other question on this. Dr. Sharanya Manoharan asked where could I get some experience in handling sequence processing and the bioinformatics analysis. Ms. Chear?
Okay, thank you. Thank you, Dr. Saranya, for the question. At this moment, we did not perform our whole exome sequencing in our laboratory yet. So I believe you can contact any service provider regarding the experience in handling the sequencing. As for the bioinformatics analysis part, we will, in our lab, we will always welcome collaboration. If you need any discussion on the bioinformatics analysis, we are always welcome you to contact us. Thank you.
Yeah. Oh, thank you, Ms. Chear. On top of that, handling sequence processing. Well, as what Ms. Chear has been mentioning, you can contact the provider, service provider, or you can also perform it in IMR, because we do have a shared facility that provides next generation sequencing. So you can also contact us, and we will lead you to the right person to handle this sequence processing for you to have training, for example. Thank you for the question. Maybe, okay, there's another question. Can you accept sample from buccal mucosa to replace the whole blood for DNA extraction for whole exome sequencing testing? Dr. Farid?
Thank you for the question, Dr. Anas Fiza. The use of buccal mucosa to replace whole blood for DNA extraction, in some other labs they do, but at the moment we are familiar with extracting DNA from peripheral blood. We will explore the possibility of using buccal mucosa because this will actually help so much, especially if the sample are from Sabah, Sarawak or remote areas. And then it will also help if we can offer this to children, for instance, because it saves them from so much of blood taking. We are interested to acquire the skills, and we hope that one day we can offer alternative blood or buccal mucosa for our patients. Thank you.
Thank you, Dr. Farid. Things to add on is that sometimes whole exome sequencing requires a quite generous amount of DNA, and based on how people extract that buccal mucosa, so it may be different, and we may not get a quality that is good for the whole exome sequencing. So we may need to explore this method, because in that case we will be able to receive samples from Sabah and Sarawak without no problem, because blood, of course, the quality, we have to think about the integrity of the samples, and there are so many other issues to think about when it concerns blood for whole exome sequencing. So yeah, that is one thing that we may want to explore. Thank you, Dr. Anas Fiza, for the question. I think I'm afraid that is all the time we have for questions from participants. So if there's any additional questions, we are happy to answer, and we will contact you through email. So we hope that this webinar has shed light on some research work that we are conducting in our institute, and would spark interest and initiate research collaborations in the future. So ladies and gentlemen, now we have come to the end of the webinar session. I would like to thank all participants for their support to make this session a successful one. You may subscribe to our National Institutes of Health Malaysia YouTube channel and click for notifications. Whenever there is a new post, we will notify you. Before I conclude, I would like to announce that we will have the next session of IMR webinar series on the 27th May 2022. The theme for next webinar is Endocrine and Metabolic Insights into Clinical, Preclinical Perspectives. So do mark your calendars. We hope you can join us the next session. Until then, many thanks from all of us in IMR. Take care and stay safe. Thank you.